Omega-3 fatty acids have earned widespread acclaim in recent decades, celebrated for their powerful anti-inflammatory effects, cardiovascular protection, and contributions to brain health. Found abundantly in fatty fish, flax seeds, and marine oils, omega-3s have often been hailed as a cornerstone of nutritional longevity. Yet, as the science of aging grows more refined, particularly in the realm of telomere biology, a surprising pattern has emerged. Omega-3 supplementation may not offer the same protective effects on telomere length as vitamin D or other targeted nutrients. Despite their broad health benefits, omega-3s seem to fall short in this one critical biomarker of biological aging. This observation arises from several randomized controlled trials and longitudinal studies comparing nutrient interventions and their effects on telomere length, which is widely considered a proxy for cellular aging. Telomeres, those repetitive DNA sequences that cap and protect our chromosomes, gradually shorten with each cell division. Their erosion is accelerated by oxidative stress, inflammation, and poor metabolic health. Nutrients that reduce these stressors theoretically could preserve telomere integrity. And yet, omega-3s appear to have a more indirect, inconsistent, or weaker relationship with this process than initially hoped. In one of the most revealing trials, researchers directly compared the impact of vitamin D and omega-3 fatty acids on telomere length over several years in aging adults. While vitamin D supplementation, at sufficient dosages, was associated with slower telomere shortening, omega-3 fatty acids failed to demonstrate the same measurable benefit, even when taken in high doses and over a long period. Why might this be the case? First, while omega-3s are potent anti-inflammatory agents, they act primarily by modifying the production of signaling molecules called eicosanoids and reducing pro-inflammatory cytokines like IL-6 and TNF-alpha. These effects can help reduce chronic systemic inflammation, which is indeed one factor behind telomere degradation. However, omega-3s may not meaningfully penetrate the nuclear machinery where telomere protection happens. In contrast, vitamin D directly regulates gene expression, influencing hundreds of genes involved in DNA repair, telomerase activation, and genomic stability. This genomic action may explain why vitamin D shows more direct telomere-related outcomes, while omega-3s operate more peripherally. Second, the bioavailability and balance of omega-3s in the body are highly variable. The two key types, EPA and DHA, serve different roles, and not all supplements are created equal. Some formulations have low absorption rates, and unless taken consistently and in adequate amounts, they may not achieve therapeutic thresholds in tissues. Additionally, the background diet of individuals, particularly the ratio of omega-6 to omega-3 fatty acids, can blunt the expected effects. A high omega-6 intake, common in Western diets, can compete with omega-3s for the same metabolic pathways, reducing their anti-inflammatory potential. Third, oxidative stress, a primary driver of telomere shortening, is not always significantly reduced by omega-3s. While these fats may lower inflammatory markers, they are themselves susceptible to oxidation. Without adequate antioxidant support, like vitamin E or selenium, omega-3 fatty acids can become pro-oxidant within the body, potentially offsetting some of their benefits. This oxidative vulnerability may weaken their capacity to preserve telomeres, especially if the individual's diet lacks sufficient antioxidant buffering. Fourth, individual variation in genetic response may play a role. Some people carry gene variants that influence how well their bodies convert, transport, or utilize omega-3s. Others may not respond robustly to omega-3 supplementation at all, a phenomenon increasingly studied in personalized nutrition. In contrast, vitamin D receptor activation appears to be more universally impactful on cellular health pathways, which may again help explain its superior showing in telomere-related metrics. None of this suggests that omega-3s are ineffective or unimportant in aging. In fact, their benefits are well documented in heart disease prevention, cognitive maintenance, and even mood stabilization. They may also reduce markers of biological aging like systemic inflammation, arterial stiffness, and lipid peroxidation, which are critical to healthy longevity. However, when it comes to the specific outcome of preserving telomere length, their effects appear more modest, indirect, or highly variable. It's also worth noting that most omega-3 trials were not originally designed to track telomere dynamics. In many cases, the dosage, duration, and participant health status were not optimized for measuring subtle changes in cellular aging. In contrast, studies focused on vitamin D have increasingly been tailored to explore DNA-level outcomes and may therefore be better equipped to detect such associations. This discrepancy highlights an important principle in anti-aging nutrition. Not all healthy nutrients target aging mechanisms in the same way. While omega-3s excel at controlling systemic inflammation and promoting cardiovascular resilience, they may not exert the same direct genomic regulation required to preserve telomere length. For those looking to build a nutritional anti-aging strategy, the lesson is to adopt a complementary approach, combining omega-3s with other agents like vitamin D, magnesium, polyphenol, and antioxidants that collectively protect DNA, reduce stress, and regulate cell replication. In closing, omega-3s remain a vital part of any longevity-focused diet, but they should not be seen as a standalone solution for halting cellular aging. Their inability to consistently preserve telomeres may reflect both their indirect mechanisms and the complex nature of telomere biology itself. Still, when combined thoughtfully with other nutrients, particularly vitamin D, they can form a powerful supportive base for aging gracefully, protecting the heart, mind, and body even if they don't directly lengthen the ends of our chromosomes.